Okay, we can get started.
Yeah.
Great. So it's really my pleasure to introduce Marco Molina for this seminar. And just a little background. So Marco actually is his BS degree was in Brazil. And after that, he had a few years experience in the industry working on transgenic crops. And in 2009 to 2013, Marty Dickman at IPGB invited him up, and he was a research associate starting the transgenic work and started in the MEPS program. And then went back actually in 2013 to 2016 with Bayer working on sugarcane, transgenic crop development, and returned a second time in 2016. And has been with us since then. He's actually doing a Ph.D. with me, as well as his full-time job, which is a research specialist with the multi-crop transformation facility. So today his topic will be talking about all the different transformation activities they do, and it's really been great working with Marco. And Myra, of course, they're experts in transformation. So it should be a pleasure hearing from Marco today. So Marco.
Hello, everyone. Thank you very much, Dr. Thompson. Very kind. And I hope everybody's listening to me well. And I will try, please bear with me. Obviously, English is not my first language, and sometimes I just go crazy here. And hopefully I don't digress too much in every single slide. But yeah, and questions are open. Please, anytime, just interrupt me. I have the tendency as well to non-stop. Many of you guys probably know that my first slide, maybe it's getting old, but we like that one. GFP is part of our validation process pretty much all the time in every experiment, in every progress that we do. It's very clear result in several steps during the tissue culture process and help us a lot to know if our things are working or not. We do a lot of protocol development. So pretty much we get protocols from the literature and try out. We try out and see if we are able to replicate it, and we see if they work or not. And GFP is very part of our daily basis of assessment, and we like it a lot. Over here, there potato, cotton, melon, and other things. Sorghum as well. Okay, yeah, as Dr. Thompson mentioned, we are coming from Brazil. I'm originally from Ecuador, but I was in Brazil since I was 7 years old. So I feel more like a Brazilian as well, and which is good. We are unable, okay. We come from, this is our beloved university back in Brazil, the ZAUC, part of the University State of São Paulo. We spend most of our years there. There's where I met my wife, and since long time ago. And meanwhile, as Dr. Thompson mentioned, the majority of our experience is coming from the industry. Those are the main groups that we work for. My last previous work was on Bayer. My wife was on CTC, it's a breeding company, sugarcane breeding company in Brazil, and other companies as well. This is our city over there as well, Piracicaba. This is where we're coming from. And as Dr. Thompson mentioned, we had the opportunity to come here first time back in 2009. Yes, 2009. This is me right in the back. It's my wife here in front. This was our first lab that we participated. This is old Dr. Marty Dickman. He passed 2 years ago. There's also beloved friends that we make along the way. This is Brett. He's back in Australia, and other people. So a long time passed between our first and second visit here to the US. And the first time we came here to do sugarcane. Sugarcane, we are known, but we know how to do sugarcane. We know how to do the transformation. Sugarcane known to be very recalcitrant, very difficult to transform. And that was our ticket to the US. Okay, you guys can do this. Just please come back and come here and do this for us. We were messing with some of Dr. Marty Dickman genes of apoptosis genes, SED9, BAG4, BAG6, some of those genes. And they were trying to test that in the abiotic stresses in sugarcane. That project was funded by Chevron back in those days. And we did a lot of work. We developed protocols for two local varieties. I don't remember names, but was very successful in the point that we were able to get maybe 1 or 2 publications out of that, 1 patent. And the patent is right now being tested by the US sugar mills and is on the research pathway right now. Actually, I need to check on them what's going on. But what's really cool, just as an example, on the top right figure, there was cold resistant, cold tolerance results. The plants on the right were the transformed plants. On the 2 on the left are controls. And at the bottom left was the drought tolerance experiments or some tests, initial tests that we did. The 2 on the left were our controls. And so apparently those genes were doing something on the transgenic lines. And we did field experiments. It was very, very nice period of our life that we spent here the first time. But then we decided to get back to Brazil back in 2016. We came back in 2016 with the mission, with the target, with the idea to come back and be part of this community of facilities here in the US. So it's not something unique, right? But we are getting there. But we are among many good, reliable sources of people and universities that have some sort of facility, some sort of services provided for the community, some more specific, some more broad approach. I know that the Florida one, they're focused on citrus transformation. Other ones are more in core. Well, depend of the community, inside community, what they are required to do. And this is what we have been doing. Okay. And we are among those very, very important people. We like to do the best of our work. We take our job very serious. We know how important this is for our partners, for our collaborators, for the PIs. We are part of your work. We are part of your project. We take that very serious, and we would like to do the best that we can. And that's a lot of responsibility, and we're ready for that. And my presentation is just to show you guys what are the things that we are doing, what things that we are capable of doing, what we have been doing so far. And we work different from other facilities that are established for longer years or more specialized in specific things. We're still working, how can I say, on demand. So depending on you, depending what is your project, depending what is your interest, depending what is your crop, depending what is your timeline, depending on you, we still are open to work with you guys. So the best that we can. And AgriLife decided to put some pipeline of facilities that provide that kind of services to the community, internal or not. As you guys know, Dr. Johnson with the Bioinformatics Service Lab, Dr. Thompson with the Genome Editing Lab as well, and we are part of that, right? We are part of we are in midway. So they figure out the genes, they do the sequencing, they do the constructs, and we are responsible to put those genes inside the plants, whatever crop it is initially. That's the ideal world, right? And again, we take that very seriously because we are part of one pipeline, we are part of a project, and our product is a transformed plant in the end of the pipeline. So if I don't deliver, nobody goes forward. So again, very serious. Everybody knows where we are located, right? We're here at Norman Bullock. From the first time and the second time, this building has all we need, and we are very well located down there. We are neighbors. Very recently, we were just added to the sowing crop science, so we are happy to be here with you all now. We do have access to the greenhouse, the grow chambers, greenhouse in our case, either in both ends. They are important both ends of our pipeline, either to generate the explants, immature embryos, leaf explants, whatever we need to do, for example, for wheat or sorghum or any kind of things, or at the end of our pipeline, whenever we have the regenerate plants and we need to grow T1 seeds, T2 seeds, or whatever the demand of that project. So we have access to that. And having those plants side by side to our lab is very important for us as well. Our lab is nothing fancy. It's nothing fancy at all. We just need a little bit of space, of bench space. Our most important requirement resource is the laminar flow. This is where we spend most of our days, not nights, because we are also following a very flexible schedule where we respect our 5 p.m., 6 p.m. Eventually, we stay longer, but we like to do the things in a very efficient way. But this is where we spend most of our time. Okay, from the past 4 years, these are the main things that we are developing right now. Sugarcane is there just because we like sugarcane. Hemp is one of the major drivers right now. We are with 2 projects going on: sorghum, onion, wheat, peanuts, cotton, rice, potato, and melon. So one of the main things that we have been exposed to during the past 4 years, we didn't decide to go for that. You guys brought that to us and with the challenge to make it happen. And that's what we are trying to do. And we are doing it in different levels, different strategies, depending on whatever is the demand. But the things that we do the most is the one in the bottom is the protocol development. Protocol development just because we don't work so much with the model crops, with the model varieties, with the model genotypes. We pretty much don't have any Arabidopsis, tomato, or tobacco in our lab. We have some, but not much. We always receive something more challenging. We always receive some non-model variety for the new crop, some commercial type of variety, some elite variety that they are known to be harder to transform, harder to put them through the tissue culture. And that's what we do. We do protocol development based on the vast literature that is available on anywhere that you look at, right? So we do a very good job in looking through the possibilities and try out that in-house. That's not my idea to explain you guys plant transformation at all. You pretty much in science and everything here, you guys are the experts in most of the things that I'm talking about here. I just want to point out that, as I mentioned, we don't work with model crops. So we usually work with the plants that are hard to do. We usually don't work with the model varieties. For example, even if we have a crop that has a variety that it's known to be easier to transform, usually that's not the ones that we work. We always bring something different, some elite variety, so that adds difficulty to the process. The seasonal explant availability. Again, sometimes we have to work with immature embryos on the greenhouse. They need to be on the right timing. If we miss 1-2 days of difference, could be the difference to have a regenerate transformer plant or not. It's very important. So that's another difficulty. Independent transformants. What I mean by that is the same what the transformation efficiency. So we need to do tons of explants, expose them to the agro or the biolistic methodology, and we expect low efficiency from those initial experiments. So this is also harder to do. We are capable of do agro and biolistics depending on whatever our partner, our client, our collaborator requires. Sometimes we go for the agro path, sometimes for the biolistic path. We are, as you guys know, we are concerned with the copy number agro. We are not concerned with that. We just want to show the initial result. We just want to show some growing GFP. We do biolistic. So again, it's tailor-made. Depending on what you guys need for us to do, we are able to do. Selectable markers also. We work with a variety of selectable markers, antibiotics, and obviously the herbicide, plenty of selection. That's something tricky as well. Sometimes we need to adjust our established protocol to a different selectable marker, maybe because the plasmid is ready for that and we don't want to mess with the plasmid. It's working for years. That's what we have to do. But whenever you introduce that variability on the tissue culture process, you can mess around with the regeneration itself. We need to do kill cuts. We need to start from zero. So it's again, another level of difficulty. And traceability, I will say, is something very important. We, as mentioned, we have several partners, we have several clients, we have several labs, we have several crops. Everything has to be managed in an organized way inside our lab, and we take that also very seriously. If I tell you that this little plant is a rice, can be something else, or this variety can be something else. And on top of everything, is known also that the majority of the projects that we are doing right now is with gene editing. Gene editing is also known to decrease that efficiency on top of everything. So we have to do, and to be honest, it's nothing fancy. We just need to do more at the beginning. Obviously, in every single step of the process, we improve little by little. For every single project, we improve our efficiency as well. Few numbers just for last year, we were messing with around 14 crops, among them sorghum, cotton, tobacco, hemp, corn, little bit of everything. They are in different stages of development. Some of them we just implemented the tissue culture only. That is pretty much just get an explant and get a regenerating plant from organogenesis or whatever, and say, okay, we are ready to work with whatever comes for that. We prepare almost 600 liters of media. We prepare around 10 to 15 liters of media per week. By now, we might be ready more close to the 1,000 liters in this period. Petri dishes, 20,000. Number of explants handled, 220,000 explants. When explants, I mean either a seed, a cotyledon, a hypocotyl, a piece of leaf that we introduce to the process of tissue culture and or transformation. One of the nice things, some improvements that we did along this process the last year was we were able to reduce in 6 months the time that we were taking to produce a cotton plant, transgenic cotton plant. We were able to reduce in 6 months. This is awesome, but it's still a long period. Literature, around 18 months. We are able to 10 to 12 months start to recover and regenerate plant. This is a big deal. 6 months is 6 months. It's still 6 months, but it's just depending on the nature of the crop. If we are talking about rice, for example, next one, rice, we did literature 3 to 4 months. Now we are doing in 2 months, 2.5 months. It's really damn fast when you compare it to cotton. And other things that we've been doing, we are optimizing peanuts protocols for herbicide. We are optimizing wheat and sorghum for bombardment. The rice I just mentioned, we have sugar beet protocols that we are in development with the USDA sugar beet and also USDA cotton. We are doing some projects for them. And for the partnership with companies, hemp, we have 2 projects going on right now with the industry, the private industry sector. They're really going well as well. I will talk about that later. There are 2 cases that we'll detail a little bit more, and hemp is one of them later on. I intend to do a quick presentation. We won't be too long. Bear with me. Just pictures of now I just want to show the process itself. I don't know how close you guys are with the tissue culture, with the specific crop. This is in the case of wheat. This is a 10-114 that we performed since when we arrived. We're very lucky with that one. We have to optimize. You see, this is the size of the immature embryo of the seed. The difference from this very immature seed from this one is night and day. And when you see the complete cycle here until we have the plant, this doesn't happen usually in our first, second, third, or fifth experiment. We just get stuck here. We never produce calite, or we never produce, the plants never regenerated. So this is part of the improvement in every single step. And this is for 10-114, is an elite variety. Its possibilities are there. We are ready to move forward for some wheat. Okay, but then you say, hey, Mark, okay, can you do this other variety, 306? We'll say, no, I need to test it out. But now we have a good base to test the new varieties, new genotypes in our very well set baseline of the transformation. And again, transformation only will occur if we have a very good base tissue culture process in place. So you need to be sure that variety will be able to regenerate. Then you add the difficulty you're exposed to the agar. Then you can bombardment. Then you can do plant selection. But if you don't have the baseline of the tissue culture, it's very hard to get the transformant in the end of the process, right? This is what we do. We make sure that we have a good base to start any single project. And pretty much this is what we have been doing in the past 4 years. 5 almost. One more for wheat, just pictures for the YFP regeneration. Just an example to show how many explants we had to expose to get everything done. Around 5,000 immature embryos bombarded. 5,000. That's a lot of experiments that were not made in one day. They were made in several batches, and every single batch we were either successful in one step or just completely dead tissue, and it's part of the nature of the process. Potato, we were able to do these 4 varieties so far, and again, we are setting up a good base, a good fundamental tissue culture transformation pipeline for whenever we have another proposal, another project, another exploratory whatever in potato we are ready to do. Potato was also straightforward. We took us some time to do, but now it's in place, it's ready to go. Whenever we have received something new, we can expose that new variety to our preset protocol development. Sorghum, same thing, very tricky, very supposed to be simple, but it's not. In this case, it's very dependable of the quality of the initial explant. In this case, they're immature seeds, immature embryos. So if your seeds are way too much contaminated on the field or in the greenhouse, you will never leave this stage only. You will never get to the tissue culture and generate calli. So it's a very thin layer between kill the explant and clean the explant. So, and we noticed that sorghum has been very sensitive to the quality of the initial explant. But we are working on that, and there's one more of the crops that we are working with. Oh, sorghum is nice to mention that we are doing the TX430 for this case. We had a project not long ago, maybe last year, 2 years ago, maybe, where we tested out the tissue culture only, just the regeneration part for 11 other varieties of sorghum with Dr. Rooney. And we were able to select out of those 11, 3 good ones that respond as good as the TX430, known as being the model variety for that crop. So now with that, we were able to get out of this model variety, maybe try something else, something different in a different variety. So this is part of our job as well, is try to find new possibilities. So I guess we did with 3 other varieties that they're maybe coming in future projects. Just an example, we had some coffee laying around over there. We had an inquiry, quote inquiry for a partner. They were thinking do coffee, and we tell them, well, we had no idea how to do coffee. We never did coffee. We are good in sugarcane, but let's try out. And we tried, and it was actually very rewarding to see the coffee coming, and it was totally new for us. But to get to this F picture here was almost 2 years. So 2 years. If you think your experiment will take longer than 6 months to do some transformation in, I don't know, maybe what is 6 months? Sugarcane is 8. I don't know. Potato, 5 months. If you think it would take too long, thinking coffee is 2 years with just tissue culture. We don't know if they're gonna actually work, but it's there, it's ready to go as well. It was fun. And obviously, most of our experiments are going through the CRISPR-based gene editing pipeline. So most of the things that we are receiving in our lab, it's maybe 80% is CRISPR. We are being successful with doing right now, so rice, wheat, peanuts, and cotton. Those are the things that we're more heavily working on with CRISPR. Those three first examples are on PDS mutant plants. This is a gene of interest for one of our collaborators. We're able to deliver 100 plants, mostly validated by either by the selectable marker or by the mutation on the gene of interest. That was really rewarding to be able to do that. And with that, my next part is to show you 2 cases of our main projects that are working right now. I don't want to go too deep on that. It's not at all my intention, just to tell you guys what most interesting things that we are doing right now. 2 weeks ago, I guess, one of our papers were published. of course, with the guidance and collaboration with Dr. Thompson here. Then this project started as like another one. Let's do some transformation in rice. Rice should be easy. Remember, Dr. Thompson, rice should be easy. And we try with all the literature available. We know the rice is really a lot of literature on that. So we say, okay, that will be something straightforward. In the end, it was so hard. And what was so hard, we said, okay, maybe we have something here that we can show to the scientific community. And we end up to publish a paper, a very straightforward paper, but very important for the transformation side of the things, how we approach our work. And again, there is a lot of literature in temperate japonica varieties, and we try a lot at the beginning and never worked. Never worked. We tried several protocols established and never worked. And we knew that something was going on with the tropical japonica varieties. In this case, variety that we were working with, the Presidio one, is very important here for the US, for Texas and South America. Most of the varieties are based on that genotype, on that background. And here in the US, still it's a known variety. Around 15 years ago, it was released, but it's still one of the most, I guess, the second most planted here around. So with that, we optimize this protocol a lot after a lot of research, after a lot of try and errors, after a lot of techniques, after a lot of variations from the original protocol that we were basing on, right? And for the validation, we opted for the gene editing strategy, trying to knocking out two genes, the PDS and the YSA genes, known to confer the albino phenotype. So, okay, if the thing is, if we are able to do the transformation, maybe we will be able to find a nice phenotype that we can easily select on the plate. That might be easier down the road to select further experiments. There's something that you can add to your gene and say, okay, maybe this plant will be easier to select down the road. And that's the initial idea, and we were able to do that. All the molecular analysis, the molecular engineering here for Dr. Thompson's lab, I'm just going to show you the pictures. But pretty much we tested out our guide RNAs for the two genes, pretty much the guide RNA1, guide RNA2, for both of them. We're just showing the template, and here to show ribonucleoprotein assay, just to show the direct cutting on the right places. So that system Cas9 guide RNA should be working. Is working, is there, so it was ready to go to send to the transformation path, what we did. And yeah, we used two guide RNAs for each. And then we performed pretty much, we used both strategies, either agro transformation and biolistic transformation for PDS and YSA. Number of explants that we tried, that we performed on during the experiments. Number of regenerated plants under the hygromycin selection plates. And here are the albino plants that were regenerating in every experiment. Okay, for the PDS was very, very noticeable that 60% of the regenerated plants show some kind of albino phenotype. And that was easy to see. And to be honest, at the initial phase, there was too obvious. So, oh no, there may be something wrong with that. We are seeing a lot of albino plants in our plates. We need to double, triple, and check because there might be something wrong. But no, indeed, most of the molecular analysis were very, very close related with the mutation on those genes, either on the guide one RNA region, or the guide RNA region, or the second guide RNA region, either the same mutation on one allele, on the second allele, or the same mutation in both alleles. We had all those kind of sort of combination of things. And out of that, around 12% of the initial explants tested were albino phenotype efficient. So around 12%. Well, it's not much. It is. Sugarcane, we are used to with efficiencies of 1%, 3%. So there was a good sign that it was working. And just to show you guys some of the variation that we have for the PDS, we have insertions, deletions, some substitutions. There's a number of plants that we recover for every single characteristic, for every single phenotype. Either on the first guide RNA or on the second guide RNA, the majority of the plants coming from this less than 10 base pairs. This is the guide RNA region, our PAM sequence, and some of the examples of the mutations, substitutions, and or deletions that we have here for those ones. So, and again, those phenotype, those molecular analyses were matching with the phenotype that we were watching on the plants. Same thing for the young the YSA. Although the YSA was not really clear much, the albino phenotype was more on the PDS side, on the PDS gene. We got the mutants, but not the phenotype as we expected. Just to show the path, the cycle of this transformation, we start from, there was another plus. Most of the protocols that were working, other than Nippon Bari and Kitaki, the most model varieties for rice, everything that was out of that was with the use of immature embryos. So, and again, the immature embryos sometimes it's hard to get the right timing. It's right to get them on the greenhouse. You have to match exactly the data you need, or maybe 1 or 2 days before or after. It's very tricky to work with immature embryos. So we were aiming for the mature seeds, calli formation, regeneration of hygromycin. And here's some of the phenotype that we noticed. This is the wild type. This is some variegate phenotype that we got, and this is some totally albino phenotype, just like the wild type plants. This is supplementary pictures just to show us as well. On the PDS side, we also got some more variegate, some dwarf phenotypes, but mostly equal to the wild type. And here the YSA is very hard to see, but if you can see in this detail here, it's young seedling albino phenotype. It's really happened when you have only 3 leaves, and other than that, whenever they get on the 6th leaf stage, they just recover. So it was very tricky to get them at the beginning. If you don't get it, you never see the phenotype. So in conclusion, we were successful on optimizing that protocol. We were successful on getting the PDS to work, and more than 60% was good enough, and we're very happy with the results. And we expect, as I showed before, we are doing kind of the same approach for wheat, peanuts, pretty much wheat peanuts, the most advanced ones, and we expect to get some kind of publication down the road with that as well. Other thing that I just want to mention very, very quick is the HAM project that we are involved with. And was one of those projects where, in this case, we have but we have very less available literature online, where you don't have a set up protocol, you don't have many years of transformation. It's something that is still ongoing. It's not really a well-defined model variety for the crop. Whenever we received this is a company project, we received kind of 5 varieties from that, different varieties, the source, and then they say, oh, try out. So we start from okay, let's try out everything we got. And this is just an example of some of the things that we try. We try cotyledon, hypocotyl, micropropagation to get in vitro leaves, calli induction. We try go direct for direct regeneration. SAM. We try different other things not shown here, but the approach was very intense. Was a very good preset matrix of combination of everything that we have been doing so far in other crops. This is one of the advantages for the facility, right? We see how other crops work, how other systems work, how other reagents work, how other light conditions work, how other temperature conditions work, and that is something that could play a role in something new, something novel that is just coming you without much of knowing. And we tried, we got it, we were able to complete at least, remember the tissue culture side first, and then we do transformation. We were able at least to do in some certain level, all of those 5 varieties, and then we picked 2 of them that were the more responsive to go forward to the transformation experiments. We were doing some GFP and GUS. Variety 1, variety 2 was very exciting to see that at the beginning, at least we are getting some transient infection, some transient transformation for those varieties. We were able to see the GUS. Those are our controls, right? A positive or negative control, just to see. This is This is all the preliminary results, and then we got excited. So, okay, let's move forward with those two. We moved forward with the regeneration. We were able to see some points here on the regenerated plants as well. Those are very tiny little plantlets on the hypocotyl side, and this on the shoots that have the GUS expression here. And then we had to optimize the protocol ongoing. I mean, we were doing this in staggered phases, and the protocol of the regeneration itself was working. Plants are looking good. They are developing in vitro. Now we are on the step to trying these things from the ex vitro conditions, just to check if they're gonna grow normally, if we're gonna be able to have T1 seeds and so on. So we're still ongoing on this project. There's very not much anything shown. And plants are rooting well in the in vitro conditions. They are going right now, going up to the soil. And now they were ready to get cut here, to get samples from the leaves. Now we're gonna go to the molecular analysis. There was maybe 2-3 weeks ago. We were able we were feel confident to clip the leaves and send them to DNA extraction and see if the gene was there or not. And luckily, yes, we got some hit. We got one hit here for this variety. As you can see here, just this one, the 14, was responsive. The plant looks much better today. They are starting developing. But for the other second variety, we have much more hits. Here we have plenty. You see, you guys can see here all the molecular results for the GUS. That was very exciting news for us, for the company, and we're gonna move forward with this variety number 2 in order to go for the gene of interest for further down the line. So they're happy, and we are happy. That was nice, and hopefully other more scientific publications or more projects that are ready to come. And that with this, I think I'm done. Just to show you guys that we do other activities here in the community as well. Scientific, we receive students, we receive postdocs, we receive researchers. Part of the groups that are working with us, sometimes they want to see and go through the process of the tissue culture. We are hands-on with whoever want to join our lab, to help and support their own project or our own projects. Other activities that we like to do as well. We don't work only. Our daughter, my son, we like to fish. We like my wife. This is my lovely wife. She likes her plants, and we are Aggies. With that, we are here to thank you guys for the opportunity. This is Marcela. Marcela worked with us. She is our colleague, also expert in transformation. All the things that we saw is run by the 3 of us. Sometimes we have work students for the semester, but the heavy load is done by Myra and Marcela. I just talk. What else? I just talk. And that's what we are here. We are here to help you. We are here to support you. This is our role. We are very proud of what we do. We know our place. We provide you guys services and whatever you guys need. Let us know how. Let us know if you need students hands-on. Let us know if you guys need pilot projects. Let us know if you need larger projects, submissions. We're here to help. This is our role. Thank you very much. Money, yes, money is very important. That was a very good point. We're a fee-based facility, right? We have to be involved in grants. We have a fee for services, and it's tailor-made. So have to reach us, and we set up the plan, we set up the strategy, and we will know. We can give you a very good estimate of how many work hours we have to do in this project. And obviously, the ones that we already have in place, they're more straightforward, more easy ones. But with that, thank you very much to everyone for the opportunity.
Questions?
But I let other people first.
Fire away.
I guess. Question. You were talking about sorghum. You said kind of the final outcome of what happens is the quality of the starting tissue, starting embryos. What controls the quality of that starting tissue?
Right. What controls
Yeah.
the effect of the quality? It's pretty much either either contamination overcome the plates, or oxidation doesn't allow the tissue to develop in cali. That's the result of that.
For example, this year, we had a lot of rain in the field, so the mature seeds that we harvested, they have some fungi growing on.
Okay,
So it was very hard for us to eliminate the contamination that came from the field. So this is something that plays a very important role for
Yeah,
us.
yeah.
Last year was much better. This year was hard.
Look, yes.
I have questions. I'm from the maize lab here. I just would like to learn what is your biggest achievement in maize corn so far, especially when it comes to in terms of the sweet culture pipeline.
Is asking about maize, what main problems with maize, corn, and the problem is we don't have a project with corn yet. That's the problem. Not the problem. That's the characteristic. We are waiting for something that we can start with. Do you know more about maize, corn? The thing is, corn, it's well known. It's a lot of literature there. That's a lot that has been done. There are few genotypes that are well known to be transformed. So if we can start with the model varieties, for the crop, the best, the better, the result will come easier or faster or more adequate. So we just need to set up a project, pilot project, anything. Oh, you're welcome. Yes. Yes, Dr. Bosselberry.
So tell me about where you got with wheat.
Well, we got wheat.
Yeah, because you were working on wheat some time ago.
Right. Wheat, the strategy for wheat was a gene editing strategy for herbicide resistance, where we got our plasmids set up in a way that they were either, the majority of them was for substitution CRISPR based, for they need a template and need to like a larger substitutions. And for that one, didn't work well. That one was, it's a random integration that never really didn't happen, either by chance or because we didn't do enough explants, even though we tried almost 2,000. 2,000? No, almost, I don't remember the names right now. So yeah, and for the knockouts actually worked. They were sent to molecular analysis, but I'm not sure if the molecular analysis was able to capture exactly the mutation that we were expecting for that specific herbicide resistant phenotype that we were looking for. So the plants were, the bottom line, the transgenic plants were regenerated and they were positive for the selectable marker. We got the mutations. We just didn't get the required substitution that we were aiming for, or either the required mutation that we were looking for the specific amino acids. Yes, please.
So my question is actually the first one is, what does exactly transformation includes? Because is it which process is, I mean, the name kind of, the first time I kind of maybe hear exactly like that, right? But where does it start? So the end is the end problem, right? But what do you mean by transformation? For me, that I'm a physicist. Yeah. Got
it. Yeah. That's a lot. But the transformation itself it's whenever we are able to express a gene in the case of the plants here, right? Whenever we're able to express a gene in a system, in our plant, our system could be any crop, as I mentioned, sorghum, wheat, or whatever. The gene of interest is the gene that we wanted to express. So that gene doesn't exist in that plant, in that system. And we want to see what will be the phenotype, the effect that the expression or suppression of that gene can do in the plant. In a simplistic way, we want to see how that gene will impact the developmental stages of that specific plant. In our case, in agriculture, we're looking for some phenotype that will be beneficial to the crop. It will be more resistant to a disease, will grow more, we got a bit higher yield production. So this is what we are looking for. So in a very rude way, this is the one we mentioned to do. The scientists, usually they know the gene that he wanna insert or put in the system, but not often they don't have depends a lot of things to make that gene insert in the plant. Yeah, one of the two methodologies that we end up with that is the agrobacterium infection. We use a bacteria system to make that gene go inside the plant, or the biolistic method that we pretty much you shoot the gene inside the cell and you expect an integration of that gene in the genome of that plant. And from that new gene insert, the the thing that we do is to make that single cell that was single or several cells that were transformed, regenerate a new plant. So that new plant that we will regenerate for the tissue culture system is a transgenic plant that came from a cell that was initially modified with a different gene.
And with the markers, so if you'd like to express any specific marker in like cell walls or in the nuclei, so this is also these are things you do.
Correct. One of the things that we checked are those markers, for example, the GFP, remember the first picture, they glow in a certain wavelength under certain conditions. We know that gene is expressed inside, so we know that experiment will work.
Okay, and the other question is related to hemp.
Yes,
So you show some plants that they seem to be in a petri dish.
right.
How big are those plants?
Can you repeat the question, Martha?
Yes, how big are the plants inside the jar for the hemp?
Or those ones, yeah,
This one's here?
yeah.
In terms of inches, 5 inches, 6 inches? The big one, the big jar is maybe 5 inches.
So if you want to be petri dish,
Oh.
It starts very tiny.
tiny is like.
Yes, like.
2-3 centimeters.
In the microscope, and then they just keep growing, but the second one, the top right, is probably less than an inch.
Oh, okay, yeah, not that big.
No.
No, oh, that's great.
Petri dish.
Because that one is petri dish, but that one is like
Yeah,
something like we
the
don't know. right?
lower picture, the jar, this jar is like a 900 ml jar, probably. The plants.
So it's not like a big. Okay.
Yeah, we're just discussing the dimensions here in the picture. So petri dish plants start from millimeters to centimeters in the plant in the petri dish. Once we transfer them to the big jars, in terms of them, it's like a 15 centimeters. So yeah, that will be ready to go to the soil. They're ready to go to the ex vitro conditions.
What do you have there? Is agarose or medium? Or what type of medium do you have there?
Yeah, agarose is one of them one of the many that we can use.
Okay.
Yeah, one of the many.
Great.
Yeah.
Looks like-
So share with them what your end product goal was with him.
I'm not sure if I can. I can.
Repeat the question.
What is our end product with hemp project? Again, I don't know if we can. That we are this is a project with a company. We are about to release a press release in the next 2 weeks, where we will express the baseline of the intention of that particular private company project. So right now, this is the most of the advance that we can share with you guys is that we are in the right path. We're getting good results. We are getting the verification that the plants are transgenic, and now we are going for the real gene of interest.
And the name of the company?
GTR. And then the second company is the Rare Earth.
Oh, rare earth.
That's the other company. We didn't start anything yet.
Oh, that's. Yeah.
Oh, you have some questions in the chat, there, Michael.
Do I?
Yeah.
The first one.
Seth was asking something about.
Oh yeah. Was asking if that's what is the variety specific.
Scroll up all the way.
Oh, okay.
Genetic map.
No genetic mapping, nothing like that. We're doing just a straightforward transformation depending on the project of the PI required for us. So nothing like that. Would you have to repeat? Okay. Comments? specific. And yeah, Bob White will be the wheat, the model variety for the crop.
Other questions?
Marco,
Yes, sir.
you mentioned there's 3 other sorghum lines. And again, repeat the question. What could you tell us, or do you remember what the other 3 potentially successful sorghum cultivars are?
Okay, the other 3 potential sorghum cultivars that we are able to identify among the 11 as a good response to the tissue culture process in sorghum. Names, I don't remember. 2019, 2928 was one of them. 2928. Oh, I'm sorry, Dr. Klein, I don't remember the name. But I remember there was something related with the parental lines that will help for the breeding process, so that we can have the transgenic lines in the parental that will not go forward. So you can kind of transfer genes through crossing. So there was,
So you got a B line.
yes, were 2 B lines. Among them were one of them was a B line that will help to do that crossing without a genetic insertion. Yes. We were very excited because something new.
And are you using biolistics for your sorghum transformation or?
We are testing both for the RTX 430. We are doing agrobacterial mediated transformation. For the 2928, we just started some preliminary tests, and agrobacterium is giving a lot of oxidation, so we also tested something hard.
Yes, correct, Dr. Rooney. That 2928 is one of them. And the question was related to that, what kind of strategy we are doing, if so it was agro or bialistic. We are trying both of them. And the agro so far has been has been a lot of oxidation to the material so far. So we're still trying. The next step is to transform maybe some report genes, some GFP or something, and we can go further. Okay, BTX 2928. BHF 25 and BTX 607.
Also, medium response is BTX 635.
And BTS 635. Yeah, among those 11, I remember there were 3 were very good. Like at the middle range were kind of, we need to do more work on that. And the bottom 3 or 4 was like, oh my God, that would require a lot. Yeah.
Other questions? Thanks for your presentation.
Thank you very much.